Bismillahirrahmanirrahim. Welcome dear students to the new lecture of histotechniques. Today we will discuss the different types of commonly used fixatives in the histopathology laboratory for the process of tissue fixation. The outlines of today's lecture is the different type of fixatives. The first one of them is the formaldehyde, which is used for the routine histopathology lab, the glutaraldehyde fixative, osmium tetroxide, methyl and ethyl alcohol, acetone and Boyne's fixative. So, these is the list of fixatives which is commonly used in the histopathology lab for the fixation of different tissues, and also these are used for the processing of tissues depending on their tests and depending on their requirements. The first one is the formaldehyde fixative. This fixative is commercially available at 40% concentration, and this 40% concentration, it is considered as a pure 100% formalin. %% of this formalin, this is used to make neutral buffered formalin, and the rate of penetration of formaldehyde or 10% formalin, that is approximately 1 millimeter per hour. So, within 1 hour this formalin can penetrate up to 1 millimeter within the tissue. The volume of formalin required for the tissues, so the amount of formalin, it should be 20 times the volume of the tissue, or in other words, the tissue should be fully immersed within the formalin for proper fixation. The mechanism of action of formaldehyde or formalin: this formalin reacts with the side chain of proteins and they forms a hydroxymethyl side groups. This hydroxymethyl side groups cross-links to form a methylene bridge. After formation of this bridge, the intermolecular and intramolecular cross-linking of the molecules occurs, and these cross-linking molecules, they ends in an insoluble product. So, the tissue is fixed in this way by formaldehyde. The advantages of formaldehyde, if it is used as a fixative, that the penetration power of formaldehyde solution, it is very high as compared to the other fixatives. The cell morphology of that is very well preserved by using this fixative, and this formaldehyde is cheap as compared to the other fixatives, and this is also one of the reason which is that's why it is used routinely in throughout the world, and it is a stable compound, that it do not reacts and hyper reacts with the other tissues or other fixatives. The solution of formalin solution is very easy to make from the formaldehyde solution, and it is the effective way of fixation for routine lab staining of the tissues. Now, what are the disadvantages of using formaldehyde as a fixative? The speed of fixation is low as compared to other fixatives, and the reaction of the formalin, it is reversible, and this formalin, it can be removed from the tissue by washing the tissue thoroughly with water, and the formalin, they cannot preserve acid mucopolysaccharides. That's why it is not used for the fixation of the mucopolysaccharides. Formalin is not good for staining of fats and enzymes, and if there is a high vascular tissue, their tissue may produce dark brown granules when fixation occurred, fixation performed within the formalin, and the exposure to the skin may cause dermatitis. It is a toxic solution, and if there is continuous chronic inhalation, they cause bronchitis in the person who is performing and who is working with the formalin. So, these are the disadvantages of the formalin solution. The second fixative is the glutaraldehyde, that is used as a fixative especially for the electron microscopy of the tissue, and glutaraldehyde, it fixes and preserves well the ultrastructure of the tissues. The fixation occurs by the glutaraldehyde is due to the extensive cross linking of the protein, just like in the formaldehyde. The mode of action of glutaraldehyde and formaldehyde is the same, that the cross linking, and this glutaraldehyde, this do not react with the lipid or carbohydrate. That's why it should be used in combination with the other type of fixatives. The penetration power of the glutaraldehyde, it is poor as compared to the formaldehyde, and only a small piece of the tissue should be used for fixation due to this poor power of penetration. The glutaraldehyde is mostly used for the electron microscopy of the tissues, and for electron microscopy, the glutaraldehyde comes commercially as 25% or 50% solutions in 10 ml, and it is used as 2.5% glutaraldehyde in 100 millimolar phosphate buffer. The pH of it should be 7, that is neutral pH. What are the advantages of using glutaraldehyde? The first one is that it fixes the ultrastructure of the tissues better than other fixative, and it causes less shrinkage of the cells. Glutaraldehyde preserves protein better than formaldehyde, and it have a good cross linking with the collagen. The glutaraldehyde is less irritating as compared to the formaldehyde. So, these are the advantages of using glutaraldehyde as a fixative for the tissue. The disadvantages of using glutaraldehyde, that it is very weak in the penetration, and the penetration power of the glutaraldehyde is very low. The tissue thickness, it should be less than 0.5 millimeter for better penetration. So, the tissue should be sliced before putting in the formal glutaraldehyde fixative. This glutaraldehyde is less stable compound, therefore, it should be used it should be used in a very quick method, in a very quick way, when it is opened, and the tissue should be directly put into glutaraldehyde solution for better fixation. It does not fixes any type of lipids, and this glutaraldehyde polymerizes above pH of 7.5. And the last one is that glutaraldehyde is costly as compared to the formaldehyde. Osmium tetroxide, this is the next commonly used fixative, and it is commercially available as 0.1 to 1 gram vial, and 2 to 4% osmium tetroxide is used in buffer of pH 7.2. Osmium tetroxide is also used for the electron microscopy of the tissues, just like the glutaraldehyde, that is also used for the electron microscopy, and this osmium tetroxide reacts with the unsaturated bonds in the lipid molecules and it fixes them. The penetration power of osmium tetroxide, it is poor, and if osmium tetroxide is used alone, then the high amount of protein and carbohydrate may be lost from the tissues by putting the tissue in the osmium tetroxide as a fixative. The advantages of osmium tetroxide, that it is very good fixative for lipids, and it preserves cytoplasmic organelles, for example, the Golgi bodies and the mitochondria. And the third advantage of the osmium tetroxide, that it does not make the tissue hard. There are many disadvantages of osmium tetroxide when it is used as a fixative. Osmium tetroxide does not fixes the proteins and carbohydrates, and it should be used in combination with the other fixatives, because it may react with the ribose group and may causes DNA clumping. Osmium tetroxide have poor penetration power, and it may also cause swelling of the tissues. If osmium tetroxide is used at room temperature, so it is toxic and it volatilizes, producing a harmful vapors. These vapors, they are toxic to the eyes and to the respiratory tract. And the last one is that it is costly, it is expensive. That is why osmium tetroxide is not used routinely for the lab, just like the formalin, which is used more commonly throughout the world. Methyl and ethyl alcohol, these are used as dehydrating agents, and they are mostly used for the fixation of the cytological smears. The tissues or smear containing water should not be put directly in the higher concentration of alcohol, because if it is put directly into higher concentration of alcohol, it may causes distortion of the cells due to the rapid removal of fluid from the cell. So, for processing the tissue in alcohol, graded alcohol should be used for the dehydration purpose. 95% of ethyl alcohol is used for fixation in the routine laboratory. Acetone is the next commonly used fixative, and it is mostly used for the enzyme studies and immunohistochemistry. It is a poor fixative for morphological preparation, and acetone is the fixative which causes significant and high amount of cell shrinkage. Acetone also works by the dehydration of the cells, and cold acetone is normally used at 4 degrees centigrade for the process of tissue fixation. The last fixative of today's topic, which is used commonly in the lab, that is Boyne's fluid. This Boyne's fluid contains picric acid, and it is excellent fixative for the glycogen. This Boyne's fluid reacts with the protein and forms protein picric acid complex. The penetration power of the Boyne's fluid is high, and it fixes small biopsies within 3 to 4 hours. And Boyne's fluid is not suitable for DNA quantitative study if tissue is to be processed for the DNA quantitative study. So, that tissue should not be fixed in the Boyne's fluid, because it damages the cell membrane and causes hydrolysis of the nucleic acids. The advantages of using Boyne's fluid: it is good fixative for connective tissue and glycogens, and it have a rapid penetration power. The disadvantages of using Boyne's fluid is that this Boyne's fluid produces yellow stain within the tissues. So, how we could remove this yellow stain, yellow color of the tissue? The tissue is first washed thoroughly with the 70% ethanol. Then yellow color can be removed by dipping the tissue in the lithium carbonate in 70% alcohol. This is the comparison of different types of fixatives. The type of fixative: buffered formalin, glutaraldehyde, osmium tetroxide. The ingredients that is present in these fixatives: in the buffered formalin, glutaraldehyde and osmium tetroxide. The advantages that formalin have high penetration rate and preserve cell morphology well. It is cheap and stable. Glutaraldehyde betterly fixes the ultrastructure. It causes less shrinkage. It preserves protein better, and it is less irritating. The osmium tetroxide, it is good fixative for the lipids and good fixatives for the Golgi bodies and mitochondria. These are the applications effective for the routine labs, and glutaraldehyde is best for the electron microscopy. Osmium tetroxide is also best for the electron microscopy of the lipids and the cytoplasmic structures. The other fixatives: ethyl alcohol, Boyne's fluid and the Zenker's fluid. These are the ingredients that insoluble alcohol, water, and the advantages of ethyl alcohol is that it have fast penetration power, but it is used for the cytological smear, and the Boyne's fluid is have rapid penetration power, but it produces yellow color within the tissues, and the Boyne's fluid is very good for the trichome stain. Boyne's fluid is good fixative for connective tissue and glycogen. And the Zenker's fluid, it is a strong, rapidly acting fixative, and it have a good penetration. The disadvantage is that the pigments are dichromate and mercury is produced, and mercury is poisonous and RBC is poorly preserved. But the application is the organ with the high vascularity such as the spleen. For that purpose, Zenker's fluid is used. That was all for the today's topic. Hope you have learned about the commonly used fixatives that is used in the laboratory. Thank you. If you have any question, you can ask through question box. Assalamu alaikum. Allah.